Hi everyone. In this video, we will explore scType, another marker-based cell type annotation method. So in the last video, we saw cluster mole, and in this we will focus on scType, and it is again R-based tool. And yep, so it comes with its own marker database, this tool called scTypeDB. So and also it uses the advantage, the uniqueness of this one is like it uses both positive and negative markers. So negative markers helps to prune false positives. So that means we want like these markers to be present, but at the same time these markers should not be present if you want to call that cell type, that kind of logic it uses. So that means that not logic means like it knows that, let's say, a plasma B cell is identified by CD138 plus, but crucially it must be CD20 minus and MS4A1 minus. So that plus and minus, right, is very important to crucially it helps us to distinguish between very similar subtypes. For example, triple negative breast cancer, right, we'll have three subtypes. They differ between ER positive, HR positive, like that, right? So positive negative markers plays a very important role, and that's the logic is used here. It allows you to construct a positive marker and negative marker. So if you already know that you have both positive and negative, and you want, then this kind of tools we can use it. Okay, and so this is the tool scType. Okay, and you can first, if you go to the database, right, they have the database as I said. So this is a database like what cell type and what marker. This is another resources for marker database that you can use anywhere, depending on the cell types and all. Okay, yep. So you see such a big database against which it will be compared. Okay, and they also have this online method by which you can upload your data. I had not tried it, but when I tried it, it took a lot of time for processing, so I did not attempt further. But you can try it out, so let it load the interface. Okay, let it load. Meanwhile, I will show you the documentation. Okay, so did it load? Yeah, so it loaded. So so you need to upload this. So either you need to upload a CSV file contains the gene expression matrix, where the rows will be the genes and columns will be the cell types, or you can upload a R object in single cell experiment object format, or directly you can pass the zip file of the cell ranger output, that matrix, barcode, those kind of thing. And you can select what tissue or what organ and what kind of data it is, normalization you want, basic log normalization you want, or SC transform based log normalization, and the visualization and additional things also you can choose, and what differential expression you want to do, whether masked or Wilcoxon, to identify the cluster differential genes across cluster, right? So it does that, but yeah, it will take some time to do. You can try it out, but yeah, they have provided this online. Okay, and the documentation is here. So they have explained here what all thing it does. The input data, it should be quality controlled, gene based. Then data normalization they do, and that they do the data clustering. Basically they follow the Surrott kind of pipeline only. And what kind of data they support, gene by barcode matrix, this kind of thing. Or you can pass the directly the cell ranger zip file you can pass. Okay, or you can pass the R object as well. And this is how the UI will look like once you will upload, and you will be able to do analysis, and you can also upload your custom marker over here, and then you can do the marker comparisons, and you will do the analysis. So whenever you have time, you can try it out. And they have the example also, they have given in GitHub link here. And this is what I have followed, and I have prepared this tutorial, but I see like it will give you more complex information if you run straight away. That's what we will be doing little more different here. Okay, so yep, that's about the scType. So what we are going to do, we are going to do annotation, take this same Surrott data, PBMC data, and we will try to annotate against their database, the scType database, their own database. We will compare that, and we will do some analysis like the extra static confidence plots, like scores, what it will generate, right? The plot and all we will see, and another heat map, we will talk about that. But at the same time, since it is against their database, you can also play around with your own database. That's what we will be doing. So this is the annotation from their database, but at the same time we will use their tool to compare against two other database, the PanglaoDB and the CellMarker. So again, now we'll have three different sources, and we will do the same approach of comparing and come to an conclusion. These three, like let's say the cluster 0 from scType database and PanglaoDB and CellMarker, and how does it look? That's what we will be doing. So I followed their tutorial and I made it little advanced to three dataset comparison kind of thing. Okay, that's what we will be doing. Okay, and this is the paper you can read. This is a 2022 paper, Nature Communication. They communicated this, and the scType does is like it's uses both positive and negative markers, and it doesn't take, that's the very important, it doesn't take only the genes. Okay, we need to provide the expression matrix. Okay, because based on that it calculates the score. And how the score it calculates, basically sum of the positive marker expression minus the sum of the negative marker. This is very simple way, but it is calculation is very different. But the simple way may it tries to differentiate positive versus negative, and it calculates the assigns the cell type. Okay, and yep, so let's do that. So I'm setting my library. Okay, so here one thing, like we need to this HGNC helper package will be needed to use some gene names, and I'm also loading my CellMarker database, Panglo database, so that we'll compare three database. And so there is no package, like, so you don't need to install dot packages, because this is not a package kind of thing. Their code is there in the this direct GitHub, so we can directly load that using the source function. Okay, so whatever the code is there, those functions are already loaded here. You see here. So these two are the functions. Okay, the one function is the gene set prepare function, using which it will prepare the genes, and this is the main function scType score, will the main scoring is happening. You see, this is very kind of very not that sophisticated, but it is using some this is the function. You can further read it. I have not gone through that what it is doing. Okay, so it has loads two function. The gene set prepare will load and process the this one for particular tissue, what tissue we want, right? And this score is the score function. Okay, and for this dataset what we will be using is like yeah, sorry, about the database, right? So their database has positive gene symbol, negative gene symbol. Okay, so means positive marker and negative marker, and the different tissue that they have available are here supported. So that's what we will be downloading, that Excel file. So let's download it. Okay, it will be downloaded in my this folder. Okay, so this is the Excel file. It's a very huge one. You see, that's what we saw on the UI, right? The same one. So here you can filter what, let's say, you want on the brain one, and what cell types and their markers. So this is the brain marker list. Okay, so you can use this one to compare, not this one. Okay, so we have downloaded it, and I think we have done that. So now let's load that, and we see that these are the different tissue that is supported. Okay, and now we need to prepare our genes. Okay, and first let's select the tissue type. For this, because PBMC we are using, like, so let's go to immune one tissue type, and this is the first function I'm calling, where I'm passing the entire data set and just filtering this immune system. Okay, so now these are the cell types that is present in our this database. So around 34 such cell type information are there. Okay, so now we will print the cell types that are having positive markers. Okay, so if I just show you this one GS list, right? So you see that this is the positive marker list, and these are the negative marker list. Okay, so positive marker list means these markers for these cells like this. Okay, and for negative means this MS4A1 should not be there in the B cell. Okay, that logic, right? And so that's why it will not be present for everywhere, so it will be blank. So only for wherever that information is there, so they have provided. So that way for immune cell, we have this kind of segregation with positive and negative markers. Okay, so now we just printing here which are the cell types that are there in the positive. So GS list dollar positive marker is this one, and this first columns I am picking it up. Let me close this one and this one. Okay, so these are the names, positive marker and negative markers. And these are the, I want to see plasma B cell, right? Plasma B cell, what are the markers are there, right? So I want to print that. So you can just say print way. So these are the positive markers for the plasma B cell, and these are the negative marker for the plasma B cell. Though we have only one B cell. Okay. So this is how we have loaded our data, their own data set, that scType. Okay. So now let's run our normal Surat pipeline to get that marker genes. Okay, this is a common thing. And okay. So now what we need is like, we need that expression matrix. Okay, so that's, and also it needs to be scaled. Okay, so that's what from Surat object under the RNA-seq we are extracting the scale data. Okay, so now this is the dimension. So we have now 2000 variable genes and 2638 cells. Okay, and scaled matrix. So you see these are the genes and these are the barcodes, and this is the scale data matrix we have. Okay, so now this matrix will pass in the first argument, and we say that this is scale equal true, and this is the positive markers all we pass and negative markers we pass. So all this positive marker and all this negative marker list we pass over here. Okay, and then this is the only function that it does. Okay, very simple. So it runs very fast to do. Okay, doesn't take much time. And here we got the result stored here, es underscore max. So here the dimension would be 30 by 2638. Okay, 30 is 30 cell types, and these are any cells. So if I just show you this one, you see that. Okay, so positive markers, right? So positive, negative, there is nothing here, but you see pro B cells for this cell type using these markers. The every barcode, it is get score was assigned. Okay, from this what we will know. Okay, this is a positive marker, right? Pro B cell is a positive marker, and we can just add all the score. And for the negative marker also it does, and based on that it calculates a difference and it will assign the final score. This is the, we call that some specificity scores they call. So now we got this 30 by means for every cell type, for every cell. Now we got the score. So this tells like, okay, see, this is my cell, and whatever is the highest, right? So this cell is probably the first one, sorry, 0.7. This is probably named CD8 plus T cell. That way you can interpret. Okay. So now these are our clusters. So what we will do now, we will assign the highest scores, right, to the cluster annotation. Some, I'm doing here some adding that information here as a just as a table, so that it will be easy for us to store the result. You see what I mean to say, like for cluster 0, and again I'm doing head, right? So okay, let's not do the head directly, we can do it, I think 30, right? So these are our 10 clusters. Okay, so for cluster 0, the first rank, the first cell type rank is this one, and that has the highest score of something was there, and we were looking here 0.73 for this cell. Let's say these three cells belongs to cluster 0. That means what you are doing for this naive T cell, you are adding this score plus this score plus this score, basically simply adding it. So that is what your rank 1 score is, first cell type score. Then second cell type, what it predicts is CD8 plus T cell, and third one is memory CD4 plus T cell. Okay, so we have 3 rank 1, rank 2, rank 3, and that's where we are calculating something called confidence gap. Confidence gap means there is a formula that I have introduced. Okay, so that is nothing but 3 minus 2 by 3. Okay, means first two, rank 1 and rank 2, the difference divided by the total, the first one. Okay, the first rank means the difference between first and second divided by the first rank score. So that means 674 minus 608 means around 70. 70 by 674, that will give us the confidence gap is 9.7. So that means more is the score, you will get more confidence. Okay, so that's the kind of logic it does. Okay, and you see now we got nice second, third rank as well. So wherever high is that, that means there is a more difference between the first and second and third rank. Okay, so that means if all the three ranks are very telling same thing, you will get more confidence and gap is very low. So further in the 33, right, highest one, let's talk about this one, right? So here you see the first one talked monocyte, second one neutrophil, third one is granulocyte. You see there is much more difference is there between the three rank. So that means confidence is kind of low over for the cluster number 7. So you can interpret that way. Okay, and yep, so that's what this information. Now what we will do, we got this information, right? What name we will do, rank 1. So that's what we will be using for the plotting. So that's why I'm using this rank 1 as a name to label it. Okay, and these are some scores. I'm storing it here, and let's do the dimension plot here. So we will get that. So now this is the naive CD4 T cell. This is NKT like T cells, but you see here we did not have very 3 clusters were there, but all of them were merged. Okay, so now what we will do, we will plot some confidence scores. Okay, basically, okay, I will just show you. And here this is the cluster 0 total score. Okay, what is the for cluster number 0, the total score for the pro beta cell. Okay, same for cluster 1, cluster 2. So higher score means it will now tell that okay, for C2 cluster B cell is like that. Okay, so this is the heat map plot. What we see, what it means, like so if you see here, this cluster 0 score is blue, is dark blue is highest score, right? So what it's like C0, this is what these are the two cells are there, and they are very close also, but that's why you see a hierarchical clustering here. And this is one group, right? This one group, these all are memory effector, CD4, CD8. So these two are one kind of, with respect to these 4 categories, we see some clustering pattern over here. Okay, but this section, if you see, right, this is more towards B cell, and that is enriched in the second cluster. So that's why oh, okay. That's why you will see that B cell is here, right, second number cluster, and for that we have enrichment here. And C4 and C5, basically C4 and C5, basically they are monocytes or other type of MPP cells. We got it. Okay, so this way also, not only dimension reduction plot, but we can also have, if you get some kind of scores from this analysis, right? So we can do this kind of metrics and heat map we can do to get the confidence. Okay, we can also do some nice bubble plot as well. This is from their tutorial only, I have picked up. Okay, little bit modification, but they are giving same information here as well. That's like yep, for cluster 0. So these are the scores. Okay, so highest score means big size means score is very high, and color means the intensity, the score positive, negative, like that. So here higher color also, right, dark. So for example, if you see 2, it's going towards B cell, right, naive B cell, memory B cell, and pro beta cell, right? And same logic, like you can see this way, means actually if you see, right, same information, this heat map and this one in a different ways we are doing it. Okay, same information in a different way. Here we are doing clustering at both the level, here we are doing in the dot plot, and the value we are highlighting here. Okay, so same information. So now next what we will do, we will do the same process, but now with against two other databases, CellMarker and PanglaoDB, and then we will see how they are doing. Okay. So that's what we will do, the custom database. Okay. So let's read the same process. PanglaoDB database says make a positive and negative. Now, since we do not have a negative marker here, for PanglaoDB doesn't give like negative marker, we can simply keep dummy. So sorry, yeah, negative is this blank one. Okay. Okay. So these are the negative one, negative we don't have, and simply you run the everything is same. Only thing like here I'm passing which positive marker from PanglaoDB. These are negative marker from this one, and rest everything will remain same. Okay. I'm just repeating the process what I've done. Okay. So now you see you will get a different result over here. Okay. So you get different results based on PanglaoDB, and now do the CellMarker one. Same process. Just repeat. So now we got little granular, right? Because CellMarker database name is, again very different. So that's why you see different output from different clusters, but the underlying tool remains same. Okay. So now you can top one hits from all these three I'm picking up and just combining them and making it wide format. Okay. So these are the top hits as per each of this database. Okay. And furthermore, you can normalize that and come up with a consensus plot. Okay, that's the next step we can do it. I have not included that part here, but you can get that from cluster mole, like how we can do the normalization, but you get an idea. Okay, so that's all about this scType database. So in short, this tool is very simple. Okay, if you go to the documentation, it's just 2 lines of code. They are also doing this kind of scoring that I have picked up from their tool, and but this is little complex way written. I have tried to simplify that, and also yep, there are more plots also you can generate by looking into the reference. Okay, the documentation. Okay, so in short, if I summarize, it uses positive and negative to do the comparison. Okay, so that's all I had here. And yeah, so last in the next video, one more final tool I will come include as well. That's EG Cell Type. This is also marker based, but this uses alternate like Fisher exact test and GAC enrichment based. It does, and it also gives different plots and information. Okay, so we will see this topic in the next video. Okay, so I hope this was very useful, and yes, so thanks, and please do provide feedback. How did you like this one, and any improvement that you need. Okay, so yeah, with that I'll stop here, and I will see you in the next video. Bye.